In this video, we're going to be looking at the muscle anatomy. This is going to be chapter 10, part 2. So muscle is really cool, mainly because it can regenerate itself. Muscle stem cells, which are called satellite cells, these actually divide during injury or development, and these produce many myoblasts. So these myoblasts, as you can see here, so these darker red ones are going to be myoblasts. They actually will fuse together to form a multinucleated muscle fiber. So here we have this, it's an immature muscle fiber, but as you can see, it's multinucleated, so it has multiple nuclei there, and it's fused from those individual myoblasts. There's always going to be satellite cells around muscle fibers, so you can see right here, and these are able to divide when they're needed to provide more muscle tissue. So the multiple nuclei of a skeletal muscle fiber are located just below the sarcolemma, and the sarcolemma is going to be the plasma membrane of a muscle cell. So here you can see that plasma membrane is going to be this blue bit. It's going to be the plasma membrane of a muscle cell called the sarcolemma. Thousands of tiny invaginations of the sarcolemma are called transverse tubules or T-tubules. And you can see them, they're labeled right here, but it's kind of these little indentations of the plasma membrane that actually goes between these sarcomeres, between the myofibrils. And these T-tubules tunnel in from the surface toward the center of each muscle fiber. So you can actually see these T-tubules here. They're going to be these lighter blue bits that go between the myofibrils right here. They go around there. T-tubules are actually open to the outside of the fiber. Because of that, they're actually filled with interstitial fluid. Muscle action potentials travel along the sarcolemma, so along the sarcolemma, and then they actually go through the T-tubules, quickly spreading throughout the muscle fiber. So as an action potential comes down the muscle cell, it actually goes in between by these T-tubules through the entire muscle fiber so that electricity penetrates the entire muscle. Now, within the sarcolemma, we have the sarcoplasm, just labeled right here. This is just going to be the cytoplasm of a muscle fiber. So although we have the sarcolemma and the sarcoplasm, they're the same as normal cells, as the plasma membrane and as the cytoplasm of a regular cell, but they're given their own name because they're part of the muscle cell, so they have this sarco prefix, just like a sarcomere. So sarcoplasm, this has a large amount of glycogen within it. And this is a very large molecule. It's composed of many smaller glucose molecules. So if we have one glucose molecule like this is just a really simple representation, but glycogen is actually lots of glucose molecules all linked together. So glycogen can be used for synthesis of ATP. So that's going to be a large reason why we have a large glycogen store within muscle cells. In addition, the sarcoplasm contains a red-colored protein, which is called myoglobin. This is a red-colored protein we see within blood cells. Within muscle cells, sorry. So this protein is only found in muscle, and it binds oxygen molecules that diffuse into the muscle fibers from the interstitial fluid. So you're going to have these myoglobin oxygen compounds. So myoglobin will bind to oxygen that comes into the muscle cell. So myoglobin will actually release oxygen when it's needed by the mitochondria for ATP production. So as you can kind of see here too, we have a lot of mitochondria within the muscle cells. As you can see right here, it's labeled down there, but you can see them all throughout here, these orange little kidney-shaped organelles. And these lie in rows throughout the muscle fiber, and they are kind of strategically placed within the muscle cells. They're close to the contractile muscle proteins that use ATP during contraction, so that the ATP can be produced really quickly whenever it's needed for contraction. So we have the sarcomeres, the contractile portion of the muscle cell, and we have a lot of these mitochondria. They're going to be placed really close to these sarcomeres so that they can produce and send ATP to the contractile parts, to the sarcomeres, when it's needed for muscle contraction. It can be a really quick process. So at high magnification, the sarcoplasm appears to be stuffed with like little threads. So these are actually called myofibrils. These are going to be these thread-like portions throughout the sarcoplasm, and these are the contractile organelles of skeletal muscle. So myofibrils, they extend the entire length of a muscle fiber. So if we have one muscle fiber here, just examine this is one muscle fiber, one muscle cell, we are going to actually have the myofibril. One myofibril is going to go from one length to the other. You do have prominent striations that actually make the entire skeletal muscle fiber appear striped. So striated is just another way to say striped. Another part of this microscopic anatomy of a muscle cell is that we have this fluid-filled system of membranous sacs that are called the sarcoplasmic reticulum. So these little sac structures at the top of this picture here, and these encircle each myofibril. So each myofibril is going to have these SRs around it. So this is kind of an elaborate system, and it's very similar to smooth endoplasmic reticulum in non-muscular cells. So this is going to be kind of the similar thing to smooth ER in other cells. There are dilated end sacs of the sarcoplasmic reticulum, and these are called terminal cisterns. And these will actually kind of butt against the T-tubule on either side. So you'll have a terminal cistern on each side of one sarcoplasmic reticulum, which will actually come into contact with the T-tubules. So you can see there. So you're going to have a transverse tubule and two terminal cisterns on either side of it will actually form what is called a triad. So you're going to have this right here is going to be a triad. So triad is made up of a T-tubule and two terminal cisterns of adjacent SRs. In relaxed muscle fibers, the sarcoplasmic reticulum stores calcium ions. So we have a lot of calcium ions within these SRs. Release of calcium from these terminal cisterns, this actually triggers a muscle contraction. So when we have calcium, we have this whole process that will actually release calcium from the terminal cisterns. This is the event that triggers muscle contraction. Now, within myofibrils, you have smaller protein structures that are called myofilaments, or sometimes they're just called filaments for short. Thin filaments are the smaller of the two. They are only about 8 nanometers in diameter, and they're composed mostly of the protein actin. Thick filaments, on the other hand, are about double in size for diameter, so they're about 16 nanometers in diameter, and they're composed mostly of the protein myosin. Both thick and thin filaments are directly involved in the contraction process, so the muscle contraction or shortening of the muscle fibers. And overall, there are about 2 thin filaments for every thick filament in the regions of overlap. So you have this region here where you have thick and thin filaments are overlapped, as well as here. You can see that these are labeled as the zone of overlap. So you're going to have 2 thin for every thick filament there. The filaments inside a myofibril don't extend the entire length of a muscle fiber. Rather, they're arranged in compartments. These compartments are called sarcomeres, and these are the basic functional units of a myofibril. So the narrow plate-shaped regions of dense protein, these are called the Z discs, and they separate one sarcomere from the next. So we can conclude that a sarcomere extends from one Z disc to the next Z disc, as we can see here. The extent of overlap of the thick and thin filaments depends on whether the muscle is contracted, relaxed, or stretched. We have a darker middle part of the sarcomere, which is labeled the A band, and this extends the entire length of the thick filaments. So toward each end of the A band, we have a zone of overlap, and this is where the thick and thin filaments lie side by side. They actually overlap one another. Next, we have the I band. This is going to be a much lighter, less dense area. You can see it labeled here. And this contains the rest of the thin filaments, but there are no thick filaments in this area. And we have the Z disc that actually passes through the center of each I band. You have a narrow H zone in the center of each A band that contains thick filaments, but there's no thin filaments in this area. A mnemonic that actually might help you to remember the composition of the H and the I bands is that the letter I is thin in appearance, so therefore we're only going to have thin filaments there, and the letter H is a lot thicker in appearance, so we're only going to have thick filaments in that area. Now, supporting the proteins that actually hold the thick filaments together at the center of the H zone form the M line. And this is named just because it's the middle of the sarcomeres. So it holds those thick filaments at the center. Myofibrils are built from 3 kinds of proteins. Contractile proteins, which generate force during contraction. Regulatory proteins, these help switch the contraction processes on and off. And then we have structural proteins, and these keep the thick and thin filaments in the proper alignment, and they give the myofibril elasticity and extensibility, and they also link the myofibrils to the sarcolemma and the extracellular matrix. The two contractile proteins in muscle are going to be myosin and actin. So these are the thick and the thin filaments respectively. So myosin is going to be the main component of thick filaments, and it functions as a motor protein in all three types of muscle tissue. So you can see this myosin molecule, a thick filament, right here. So you have motor proteins pull various cellular structures to achieve movement by converting chemical energy in ATP to the mechanical energy of motion, and that's the production of force. Each myosin molecule is kind of shaped like two golf clubs that are twisted together. You have the myosin tail, which kind of looks like the twisted golf club handles, and this points toward the M line. And this is, remember, the center of the sarcomere. And then the two projections of each myosin molecule, these are called the myosin heads, kind of like the golf club heads. Thin filaments are anchored to Z discs. And their main component is the protein actin. You can kind of see, so in this one, you can see a thin filament. You have these actin molecules, which are going to be these kind of yellow balls, as you can see in this picture. So each individual actin molecule join to form the actin filament, and this is twisted into a helix, as you can see in this picture. On each actin molecule, you have a myosin binding site. And this is where the myosin head can actually attach to. So it's this little bit like these darker portions that you can see on the actin. Then we're going to talk about these regulatory proteins. So you have smaller amounts of tropomyosin and troponin, which are also part of the thin filaments. In relaxed muscle, myosin is blocked from binding to actin because these strands of tropomyosin are actually covering the myosin binding sites on actin. So you can see it's going to be this orange fiber looking thing here is tropomyosin, and you can actually see it's covering those binding sites on the actin so that myosin can't actually bind to actin. The tropomyosin strands are actually held in place by troponin, which are going to be these blue looking molecules here. When calcium binds to troponin, it undergoes a change in shape, which moves the tropomyosin away from the binding sites on actin. And then a muscle contraction can subsequently begin as myosin binds to actin. We will go over the actual contraction process in future lecture videos. But that's the basis of how myosin will bind to actin and how in relaxed muscle actin is actually covered up by tropomyosin and troponin so that myosin can't bind to actin. Now some structural proteins. There are about a dozen or so structural proteins in muscle tissue which contribute to alignments and stability, elasticity, and extensibility of myofibrils, but we're just going to talk about a few of the key structural proteins today. First off, titin. The word titin means gigantic. This is the third most plentiful protein in skeletal muscle. Each titin molecule connects a Z disc to the M line. Connects a Z disc to the M line of the sarcomere, and this helps to stabilize the position of the thick filament. Stabilized position of the thick filament. The dense material of Z discs contains these molecules called alpha-actinin. And this binds to the actin molecules of the thin filaments and binds them to titin. So this binds the thin filaments to titin. Molecules of myomesin actually form the M line. And the M-line proteins bind to titin and they connect adjacent thick filaments to one another. So they will bind. So these we just think of these as these connecting thick filaments, these adjacent thick filaments together. Pretty much myosin holds the thick filaments in alignment, or sorry, myomesin rather, holds the thick filaments in alignment at the M line. Next, nebulin. This is a very long, non-elastic protein, and it wraps around the entire length of each thin filament, and it helps to anchor thin filaments to these Z discs and also regulates the length of thin filaments during development. So this is going to be very largely a developmental protein, helps to make sure everything develops in the right length. And then it also helps to connect everything, or connect the thin filaments to these Z discs. Now, dystrophin. I may argue that dystrophin is one of the most important structural proteins in a muscle fiber. Now, this links the thin filaments of the sarcomere to the integral membrane proteins of the sarcolemma. And these integral membrane proteins are attached to the proteins in the connective tissue of the extracellular matrix that surrounds the muscle fibers. So if we look at kind of a cross section of a muscle fiber, kind of think of dystrophin here. We're going to have these integral membrane proteins. And then we're going to have dystrophin that would actually form that connection point of the integral membrane proteins to the thin filaments. Imagine these red are going to be the thin filaments that are actually going to be contracting in the muscle fiber. Essentially, dystrophin connects the sarcomere that's contracting within the muscle fiber to the sarcolemma. Remember, the sarcolemma is going to be the plasma membrane of a muscle cell. So this bit here is going to be the sarcolemma of the muscle fiber. So if we look at it like this, we have, for example, a muscle cell here. And this is going to be the sarcolemma. And here's going to be another. So we have two individual muscle fibers here, and on the one on the left, we have, just for simplicity's sake, we have one sarcomere. Now, if we didn't have dystrophin, I'll actually go here. If we didn't have dystrophin connecting the sarcomere to the sarcolemma, the sarcomere would just kind of contract within the muscle cell and nothing would actually happen. So we'd have contraction, so this would actually get smaller, it would contract, but as you can see, the muscle fiber stays the same size, nothing happens. We require dystrophin to make that connection between the sarcomere and the sarcolemma. So now that we have that dystrophin connecting everything, now when the sarcomere contracts, it's also connected to the sarcolemma, so the entire cell is all going to contract and all going to get smaller along with that, along with all the contractile proteins, as you can see here. So that is what dystrophin is going to do. It's going to make sure that everything is connected to the plasma membrane so that the entire cell will contract together. And that's going to be the end of Chapter 10, Part 2.